Hello, everyone, and welcome to today's live broadcast, Enrichment and Analysis of Rare Antigen and Virus-Specific T Cells, presented by Dr. Marcello Stein, the Global Product Manager of T Cell Immunology at Miltenyi Biotec. I am Tracy S. Martin of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Miltenyi Biotec. For more information on our sponsor, please visit miltenyibiotech.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Marcello Stein. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Stein, you may now begin your presentation.
Welcome everybody to our webinar, Enrichment and Analysis of Rare Antigen and Virus-Specific T Cells. In the next 45 minutes, you will learn about Miltenyi Biotec's extensive antigen-specific and virus-specific T cell portfolio, including relevant application data and workflows. So enjoy. Let's begin with a brief company presentation. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi himself, and is the largest owner-managed company in Germany. We have two main sites in Germany, one in Bergisch Gladbach, where the headquarters are located, and one in Teterow, where the GMP manufacturing facility is located. We have subsidiaries in 28 countries and additionally distribute in 73 more countries. Currently, we employ over 3,000 employees worldwide. Our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. In the current situation with the COVID-19 pandemic, it has never been more important to have powerful tools at your disposal for an efficient work with antigen, or better in this case, virus-specific T cells. SARS-CoV-2, as cause of the novel respiratory disease COVID-19, exhibits high infection rates and number of cases worldwide, with partially severe causes of disease and even fatalities. This has severe social and economic consequences. So we need to act now. Miltenyi Biotec does its best to support your virus-specific T cell research. Let's have a first look on how we can define antigen-specific T cells. Of course, all T cells are antigen-specific. However, the term hereby is applied to T cells that were activated prior to analysis via their cognate antigen and not via an artificial polyclonal stimulus. Hereby, only T cells of the given specificity are activated and clonally expand. The initial frequency of a T cell of a certain specificity is, however, very low. Analysis of antigen-activated T cells is important to understand the very basics of the immune response and plays a pivotal role not only in infectious diseases, but also in tumor immunology, autoimmunity, allergology, and many more. A little more detailed look is depicted on the right side. Upon antigen-specific activation, T cells clonally expand, upregulate activation markers, and release cytokines. Thus, assessment of those markers and cytokines as well as proliferation are valuable tools for the analysis of antigen-activated T cells. Some molecules are of outstanding importance when it comes to T-cell activation and serve as good activation indicators. CD69 as an early pan-T-cell activation marker. CD25, the IL-2 receptor, as later activation marker on all conventional T-cells, but also as regulatory T-cell marker. CD137, also called 4-1BB, a suitable CD8 positive conventional T-cell activation marker, but also as early activation marker on regulatory T-cells. And CD154, also called CD40 ligand, as ideal conventional CD4 T-cell activation marker with a very low background. And finally, of course, cytokines for the functional analysis of the antigen-activated T-cells. Generally, analysis methods for antigen-specific T-cells can be classified into two categories. First, the direct analysis methods. Labeling of the specific TCR using recombinant MHC peptide multimers identifies specific T-cells directly according to their specific antigen receptor without restriction to a certain functional parameter. Hereby, Class I MHC peptide multimers are widely used to quantify and characterize antigen-specific CD8 positive T-cell responses. The construction of Class II multimers for CD4 T-cell analysis is more difficult due to differences of the MHC structure and TCR affinity. However, recently some advancements have been made in this field. Still, this method is more frequently applied to CD8 positive T-cell analysis and lacks somewhat CD4 T-cell applicability. However, the major limitation of the tetramer or multimer technology is that the antigenic epitope has to be characterized in detail. That is, a defined peptide restricted to a particular MHC haplotype. Second, we have the indirect analysis methods. This category employs functional parameters such as expression of aforementioned activation markers and cytokines, but also cytotoxicity and proliferation as an indirect readout for T-cells which react to a specific antigen challenge. The detection of antigen-specific T-cells by functional parameters requires the prior in vitro stimulation with the specific antigen. For stimulation, peptides, peptide pools, proteins or lysates can be used, covering hereby all possible T-cell epitopes. The advantage of these activation methods is that they are independent of MHC alleles or exact definition of the antigenic epitopes. However, the assays depend on the presence of a sufficient number of functional antigen-presenting cells and per definition exclude energetic T-cells from the analysis. Most importantly, the biggest advantage here is that all relevant information can be obtained using flow cytometry-based assays, and selective markers allow for additional cell isolation or enrichment. In the following slides, we will especially have a look at the indirect analysis methods for antigen-specific T-cells, to have a complete view on the functional characteristics of these interesting cells. However, as advanced the different approaches for the analysis of antigen-specific T-cells are, there are quite some challenges that need to be faced. The most difficult challenge, of course, is the rarity of T-cells of a given specificity. Depending on the individual experience of the donor, as well as of the antigen itself, frequencies can vary between 0.1% down to 0.001% of the whole T-cell population. In addition to that, sometimes those rare cells are difficult to isolate, for example when working with solid tissue. Also, it needs to be determined which cytokines and markers one wants actually to look at. Here again, the specific circumstances and goals need to be taken into consideration. We will have a closer look at these challenges, learn about individual experimental solutions, and have examples of successful applications. In the end, it is indeed like finding the needle in the haystack. As outlined before, the frequency of antigen-specific T-cells can vary depending on the circumstances and is one of the biggest challenges for the analysis. All the more, it is important to optimally prepare the starting material without losing precious cells. Furthermore, only a low background enables to efficiently find the desired cell population, as it helps to drastically increase sensitivity in detection. Of course, specific labeling, no matter if for the cell enrichment or flow analysis, is essential to lower the background. To efficiently isolate, detect, and analyze antigen-specific T cells, an optimized procedure during every step of the workflow is essential, from sample preparation to cell culture to enrichment and finally to flow analysis. Miltenyi Biotec combines different technologies over the whole workflow to maximize success when working with rare antigen-specific T cells. The whole workflow, of course, should be optimized, and the right product combination is the key. However, why are especially T cells so fundamental for an efficient antivirus response? Apart from the innate immunity and B-cell mediated antibody response, T cells play a central role in the body's antivirus defense, for example by killing of virus-infected cells directly, but also by supporting the B-cell response. All these parts of the human immune response are complementary for an efficient antivirus immune response. Consequently, only assessing antibody serology does not give the full picture. The virus-specific T-cell response has also to be analyzed. From infectious diseases and viruses like MERS and SARS-CoV-1 that are closely related to COVID-19, we know of the important role of virus-specific T cells for an efficient antivirus immune response. As Zhao et al. State in Science Immunology, as well as other recent publications, the role of T cells in coronavirus-related infectious diseases is essential for the analysis of the population's immune status in epidemiological studies, the prognosis for disease progress and treatment options for virus infections, for long-term protection provided by virus-specific memory T cells, as a target point for vaccine development, and finally also for monitoring the efficacy of a vaccine. Please follow the links on the upper and lower right side to find the respective publications in PubMed. However, to analyze the rare virus-specific T cells efficiently, several parameters have to be fulfilled. Sensitivity for the most precise results, speed for higher throughput, simple procedures for a lower error rate, a multiparameter analysis for all crucial factors, and reproducibility for higher reliability. And Miltenyi Biotec has of course the fitting solutions. So let's have a look at the in vitro workflow when analyzing antigen-specific T cells. First, PBMCs, including T cells, are isolated from a donor sample. However, in any case, antigen-presenting cells need to be present to prime the T cells with the given antigen. Within the PBMC context, this is already the case. Consequently, when working with isolated T cells, added antigen-presenting cells have to be co-cultured. Next, the cells are stimulated with the antigen of interest, for example using virus peptide pools. After a stimulation phase of several hours, the antigen-specific T cells get activated, upregulate certain surface markers, and secrete functional cytokines. Now, cells can be stained intra- and extracellularly to assess phenotype on cytokine expression. Additionally, secreted cytokines can be analyzed in the cell culture supernatants. Before the analysis, rare antigen-specific T cells may be pre-enriched. Finally, cells can now be analyzed via flow cytometry to assess the quality and functionality of the antigen-specific T cell response. A successful experimental workflow starts with efficient sample preparation. The most widely used source for antigen-specific T cells are PBMCs. They contain both the antigen-specific T cells as well as the antigen-presenting cells, and thereby provide a functional immunological context for the analysis of antigen-activated T cells. Usually, PBMCs are prepared via density gradient centrifugation. However, when working with, for example, compromised samples, contaminated samples, very small samples, or very rare cells, density gradient centrifugation can be very tedious, inefficient, and even dangerous. Here, recovery of every cell counts. With the PBMC isolation kit, small blood volumes ranging from 1 to 10 milliliters can be easily processed within 25 minutes to obtain highly pure PBMCs with a high recovery. The MaxPrep PBMC isolation kit eliminates user-dependent inconsistencies and performs reliably for every separation and is a very valuable solution. Please follow the link in the subheadline to get the respective product page. However, under certain disease conditions, tissue-infiltrating antigen-specific T cells might be of interest. In this step, valuable cells can be easily lost due to inefficient tissue dissociation or harsh digest conditions. Especially with the rare cells, again, every prepared cell counts. Our hallmark product here is the GentleMax tissue dissociator. The gentle dissociation preserves cell epitopes and recovers efficiently precious immune cells, even from hard-to-process tissue samples. Being programmable, the performance is highly reproducible. It is fast, since with the GentleMax up to 8 samples can be processed in parallel. With the heaters, you can control the temperature for optimal performance. And finally, with the sterile and closed tubes, you work in a safe and closed system. The perfect addition to the GentleMax are our tissue dissociation kits. They combine enzymatic treatment with the GentleMax mechanical dissociation. T cells can be isolated with ease, even from difficult human and mouse tissue, such as spleen, tumor, lung, liver, skin, brain, and lamina propria. The combination of the GentleMax dissociator and the tissue dissociation kits guarantee high yield, best reproducibility, and most gentle dissociation for the preservation of epitopes and thereby optimal recovery of rare and delicate cells. Please again follow the link in the subheadline to get to the respective product page. After preparing target cells, for example PBMCs, a crucial step in the workflow is the antigen-specific stimulation of the T cells. Hereby, the most efficient antigen-specific stimulation of T cells is achieved reliably with our Peptivator peptide pools. These extensive panels of virus, tumor, fungi or microbiota-specific antigens consists of 50 mer peptides with 11 amino acid overlaps, covering the complete sequence of the respective antigen. The peptide composition ensures the optimal stimulation of both CD4 and CD8 T cells. Moreover, Miltenyi's peptivators are water-soluble and do not have to be diluted in harmful DMSO. Peptivators are available in different quality grades, as well as in a convenient 96-well format for high-throughput applications. The different quality grades include research grade, premium grade, and even max GMP grade. Most notably, we recently added SARS-CoV-2, protein S, protein N, and protein M peptivators to our portfolio to support your research best in these difficult times. Please follow the respective links to get to the Peptivator product pages. As listed here, the many specificities of our Peptivator peptide pools cover the most important antigens ranging from various viruses such as CMV and EBV, of course also including SARS-CoV-2, to, for example, GP100, Melan A, to Candida albicans, as well as to autoantigens like insulin. On the right side, you can see how efficiently Peptivator peptide pools stimulate CD4 and CD8 T cells, hereby assessed by interferon gamma expression after stimulation. Peptivators HT provide quality synthetic peptide pools to enable high-throughput applications. Assemble your customized assay by simply combining modular antigen-specific strips on a single plate. This level of absolute flexibility leaves you in full control of your experimental setup. A control strip can also be easily added. More flexibility, greater convenience, and comprehensive support from the experts in T cells. Of course, flow cytometry is the most widely used method to analyze antigen and virus-specific T cells. However, even an established method like flow cytometry has its limits and challenges, as antigen-specific T cells can be extremely rare and evade detection. As described by Bacher et colleagues in Current Opinions of Pharmacology in 2015. Despite its high sensitivity, standard flow cytometry is limited by the number of events which can be acquired, typically around 1 million cells, as well as the staining background, which is typically between 0.01 and 0.1%. This restricts the analysis to populations occurring at frequencies lower than 0.01% to 0.1%. In absence of acute infections, frequency of antigen-specific memory cells can typically be in the range of 1 cell within 100 to 1 cell within 100,000. However, many functionally important T cell populations occur at much lower frequencies. For example, T cells specific for certain viruses, autoantigens, tumor antigens, environmental antigens, or neoantigens are typically below 0.01%. They cannot be assessed without additional measures such as pre-enrichment and reduction of the flow background. So, of course, ideally use max microbeads for the pre-enrichment. Moreover, to keep background during flow analysis to a minimum, specific labeling and bright fluorophores are a prerequisite. The use of reaffinity recombinant antibodies and our bright proprietary bio-dye conjugates maximizes these efforts. Later, we will hear more about that. You can also follow the link in the 2 last bullet points to get more info about reaffinity recombinant antibodies and our biodyes. So, what makes our MicroBeads MAX technology so special and the ideal tool for the pre-enrichment of rare and fragile cells? The gold standard, so to say. Under the motto, select the best, we describe the advantage of our column technology. Minimal labeling with no effects on cell biology for full downstream compatibility. The column amplifies the magnetic field 10,000-fold. This allows for minimal labeling in smallest bead size. In consequence, less epitopes are covered on the cell surface and less stress is applied on the cells. The column matrix allows for the most gentle sorting, since there is a large space between the steel spheres, around 20 times the size of a single lymphocyte. And thousands of publications agree. And on the right side, you can see the consequences. The more left panel shows column-free technology with massive overlabeling and bead aggregates. The panel more on the right shows max column technology, minimal labeling and smallest bead size. If you like to learn more about max cell separation technology, please follow the link above. And indeed, the results with our enrichment CD154 and CD137 MicroBead kits are striking. PBMCs were stimulated to CMV PP65 peptivators or left untreated. And then antigen-activated T cells were enriched using our CD154 and CD137 MicroBead kits respectively. Cells were then counterstained for CD4 and CD8. As expected, hardly any cell expressing CD154 or CD137 can be found in the unstimulated samples, regardless of enrichment. After stimulation, however, antigen-specific T cells heavily upregulate the respective markers and can thereby be easily enriched for any downstream analysis. With the CD137 and CD154 MicroBeads, Miltenyi Biotec offers the ideal tools to pre-enrich activated antigen-specific T cells. Highest purity and recovery are ensured. With the minimal labeling, full compatibility to all downstream applications without effects on the cell biology is ensured. Of course, the T-cell related cell separation portfolio is not limited to CD154 and CD137, but comprises solutions for the positive and negative isolation of several T-cell subsets, including CD4 T-cells, CD8 T-cells, and Treg cells. Make sure to have a look at the respective technology paper by following the respective link on the lower right side. In this context, the rapid ARTE protocol is the perfect solution. It combines surface marker staining with intracellular cytokine staining, but includes also an enrichment step based on CD154 expression for even the rarest antigen-specific CD4 positive T cells. First, PBMCs are stimulated with a specific antigen for some hours. The T cells get activated and upregulate accordingly activation marker CD154 and release functional cytokines. Cells are then stained for further surface markers, including CD154. Next, cells are enriched via CD154 using MAX columns, and further intracellular staining, for example for cytokines, are performed directly on the column. Then the enriched and stained antigen-activated T cells can be eluted and easily analyzed via flow cytometry in, for example, on our MaxQuant analyzer. This approach is not only very fast with the staining directly on the column, but also extremely sensitive due to the enrichment via CD154. CD154 is, as described before, an activation marker that is ideally suited to enrich rare antigen-activated CD4 positive T cells. Rapid ARTE may be modified for CD8 positive T cells by using other markers, for example CD137 for enrichment. In this example here that you can see on the right side, as described by Bacher et colleagues, PBMCs were stimulated with aspergillus lysate for 7 hours, stained for surface markers and cytokines, and enriched based on their CD154 expression. The protocol enriched antigen-specific T cells up to 85 times and thereby facilitated a reliable analysis of aspergillus reacting T cells. Rapid ARTE is the fastest and easiest solution for the analysis of even the rarest of antigen-specific T cells, with frequencies below 0.01% of total T cells. Please also download the application note Rapid ARTE on our T cell application page by following the link in the upper right side. However, let's have a look at a virus-specific example where the ARTE protocol was applied. PBMCs from COVID-19 survivors and healthy donors were either stimulated with a mix of SARS-CoV-2 peptivators, a mix of influenza peptides as control, or left untreated. As seen in the middle panel, the very rare SARS-CoV-2 specific CD4 positive T cells were successfully enriched via CD154 and visualized in flow analysis. The data was provided by the courtesy of Alexander Scheffold and Petra Bacher from the UKSH in Kiel, Germany. Furthermore, I want to point out a cell separation product that is absolutely unique: the cytokine secretion assays. They allow to detect and enrich viable cytokine secreting cells from mouse and human samples. Cytokines are immobilized on the surface of the secreting cells by the so-called catch reagent. Basically, thereby the cells label themselves with its own cytokines. In a second step, the immobilized cytokines are then labeled via fluorophore conjugated antibodies, called detection reagent, and can then be detected viably without intracellular staining in the flow cytometer. In an optional third step, the fluorophore-labeled cytokine-secreting cells can be specifically enriched and used in downstream assays by using an anti-fluorophore magnetic microbead. Consequently, our cytokine secretion assays come in two versions. For flow detections only, we have the detection kits. For the separation of viable cytokine-secreting cells, we have the detection and enrichment kits, including the additional enrichment step via the MAX column. As you can see, the cytokine secretion assay detection and enrichment kits work very efficiently to enrich viable virus-specific T cells based on their cytokine secretion. PBMCs of a CMV-positive donor were stimulated for 4 hours with CMV lysate. The responding cells were then stained and isolated according to the secretion of TNF-alpha and interferon gamma using the respective secretion assay cell enrichment and detection kits. As you can see, in the stimulated samples, a strong population of cytokine-secreting CD4 and CD8 positive T cells respectively was enriched. In the unstimulated control sample, however, no cytokine-secreting T cells were detected after enrichment. As briefly mentioned before, talking about flow analysis of rare antigen-specific T cells, one of the most important aspects is the minimization of background. Here, the reaffinity recombinant antibodies are the ideal tool. Being recombinantly engineered, we have complete control over the sequence and guarantee highest purity and lot-to-lot consistency to significantly reduce background. Hereby, the Fc receptor binding site was mutated, so no unspecific Fc binding occurs and tedious FcR blocking can be completely omitted. Combined with our super bright proprietary biodyes for clear population discrimination, you have the ideal and most precise tool for flow analysis of rare cells. Please again follow the respective links to find more product information as well as our useful tool, the Antibody Panel Builder, to design well-planned complex multicolor panels with optimized flow cytometry antibodies. However, we do not only look at antibodies, but also have ready-to-use flow analysis kits ranging from pre-mixed antibody cocktails to full-fledged in-depth kits. The Rapid Cytokine Inspector combines extracellular T-cell markers staining with intracellular cytokine staining for the analysis of activated CD4 and CD8 positive T cells. The MaxPlex assays are designed for determining concentrations of up to 12 soluble analytes in a single sample. For example, secreted cytokines in cell culture supernatants. And last but not least, the 8-color immunophenotyping kit human allows from the evaluation of various leukocyte subsets in one kit, for example, monocytes, neutrophils, eosinophils, T, B, and NK cell populations. And of course, all of these kits are compatible with the fast and convenient express modes on the MaxQuant analyzers, automating analysis and thereby not only speeding it up, but also eliminating human errors. However, we will later hear more about that. Make sure to have a look at the respective promo video by following the respective links in the right side. Let's have a closer look at some of these flow kits. For the mere flow staining without enrichment of antigen-specific T cells, the Rapid Cytokine Inspector is the ideal all-in-one kit for the combined extra and intracellular staining of antigen-specific T cells. Thereby, subtype and activation-specific surface markers as well as intracellular cytokines are stained at the same time. The Rapid Cytokine Inspector is ideal to monitor antigen-specific T cells with frequencies higher than 0.01% of the total T cell population. As said before, for lower frequencies, the Rapid ARTE protocol, including the enrichment step, is recommended. Applications for the Rapid Cytokine Inspector include assessment of samples' immune status or testing of leukapheresis material for virus-specific CD4 and CD8 positive T cells. On the right side, you can see how the Rapid Cytokine Inspector was compared to standard intracellular staining protocols. After the stimulation with CMV peptivators PP65 and IE, frequencies of CMV-specific CD4 and CD8 T cells were analyzed in PBMCs. Thereby, for example, the frequency of virus-specific T cells in leukapheresis products, as said before, can be assessed. The rapid cytokine inspector performed as good as the classical intracellular staining, but yields the benefits of a much faster protocols and less washing steps and thereby less cell loss. As said before, the rapid cytokine inspectors is of course compatible with the express modes for automated data analysis on the MaxQuant analyzers. This example shows the successful analysis of antigen-specific CD8 positive T cells producing interferon gamma and IL-2 after peptivator stimulation. Human PBMCs were incubated with or without peptivator CMV PP65 for 6 hours and brephylatin A was added after 2 hours. The cells were then stained using the rapid cytokine inspector for expression of CD8, interferon gamma, and interleukin-2. Expression was then assessed with the MaxQuant Analyzer 10. As you can see, the stimulated CMV-specific CD8 positive T cells nicely expressed interferon gamma and IL-2 upon activation. And finally, we'd like to show you an example where CD3 positive T cells were assessed for interferon gamma expression after stimulation with our SARS-CoV-2 peptivators. A blood sample was lysed with an RBC lysis buffer and stimulated for 4 hours with the respective peptivators or left untreated. Subsequently, cells were stained using the rapid cytokine inspector-based staining protocol. Interferon gamma production by CD3 positive T cells was assessed. The plots clearly show that COVID-19 survivors show elevated level of SARS-CoV-2 reactive T cells after stimulation with the respective peptivators. Another important aspect of antigen-activated T-cell analysis is the analysis of secreted functional cytokines. Here Miltenyi Biotec offers, as briefly mentioned before, flow analysis kits for multiparameter analysis of soluble cytokines in cell culture supernatants, the MaxPlex cytokine kits. They are designed for determining concentrations of up to 12 human soluble analytes in a single sample. The analysis is based on MaxPlex capture beads, which display defined fluorescent properties and can be identified using any multicolor standard flow cytometer. Again, in combination with the express modes on the MaxQuant analyzers, however, the MaxPlex cytokine kits are optimized for automated measurements and simplify flow cytometric analysis via predefined experimental settings as well as acquisition and analysis templates. Furthermore, additional variants of the MaxPlex kits, for example for mouse samples, exosome analysis, or the analysis of cytotoxic T and NK cells are available. So the MaxPlex assays allow real multiplex assays for secreted cytokines using any standard flow cytometer. Make sure to have also a look at the respective webinar by following the link on the right side. Here we have an example how MaxPlex analysis using the MaxQuant Express modes look like. PBMCs were stimulated with CV, peptides and a supernatant analyzed for cytokine secretion. The fast and easy Express modes automate analysis of unknown samples without user-dependent variations and result in most reproducible analysis. As you can see here, within minutes, a graphical representation as well as a statistical display of the measured cytokines is emitted by the software. So, what are these Express modes exactly? Express modes are software modules on the MaxQuant analyzers that automate flow analysis with standardized algorithm-based data evaluation. Results become more standardized as operator variability is minimized due to a lack of manual intervention and reduction of human error. This results in highest reproducibility with identical flow analysis independent from the operator. Express modes are simple to use and offer quick and robust analysis for easy integration into SOPs with their validated automated analysis concept. Several of our flow kits, such as the aforementioned Rapid Cytokine Inspector, Max Black Cytokine kits, the 8-color immunophenotyping kit, as well as specific flow panels, for example for CAR T-cell analysis, are compatible with the express modes. Again, make sure to have a look at the respective webinar by following the respective link in the middle of the page. In this context, let's have a brief excursion to our flow instruments from the MaxQuant series. First, we have the MaxQuant Analyzer, powerful benchtop flow cytometers with up to 16 optical parameters. The clue here is that they allow for full automation, including acquisition using a robotic sample collector with the express mode software modules. Even the data analysis is automated and free of human error. Second, we have the MaxQuant Taito cell sorter. Thanks to microchip sorting using a microvalve, the MaxQuant Taito allows for the most gentle cell sorting and highest viability, even for rare and fragile cells. Furthermore, the closed and sterile cartridge system makes the Taito the ideal machine for a GMP environment. It is worthwhile mentioning that, of course, safety in this kind of research comes first. The small benchtop cytometers fit easily under the laminar flow cabinet, and the Taito cell sorter works without internal fluidics and applies a sterile and closed cartridge system for maximum operator safety. Here I'd like to have a closer look, especially at the MaxQuant Taito cell sorter. With its unique capabilities, namely the most gentle sorting and the closed sterile cartridge system, the Taito is the ideal machine for a GMP clinical environment, not only in the fields of CAR T cells and Tregs, but of course also in the field of antigen-specific T cells. So let me point out that the Taito is already part of several clinical cancer trials involving antigen or better tumor-specific T cells, as you can see in the list on the right side. Of course, I want to share an application example for the sorting of CMV-specific CD8 positive T cells using the Taito cell sorter. PBMCs from a CMV seropositive donor were labeled with CMV dextramer PE and CD8 APC and sorted with the MaxQuant Taito. The dot plot shows pre-PBMCs prior to sorting in the input chamber, the CMV dextramer positive target cell fraction after sorting in the positive collection chamber, and the cell fraction depleted of the CMV dextramer positive cells in the negative collection chamber. Flow cytometry analysis was then performed on the MaxQuant analyzer. As you can see, CMV-specific CD8 positive T cells were gently sorted with highest purity of almost 94%. Make sure to have a look at the respective application note and scientific poster by following the respective links on the right side. To this end, here you see a schematic overview of our whole antigen-specific T-cell portfolio and workflow. From sample preparation to cell separation to cell analysis, we cover the whole antigen-specific T-cell workflow. Please make sure to have a look at our respective application website for the virus-specific T cells, as well as our webinar by following the links below. To sum up, the most important message is that we will manage this virus crisis together. T cells play a fundamental role in the antivirus immune response, and their analysis in this context is very important. There are a lot of benefits from an efficient virus-specific T-cell research. Apart from a better understanding of the antivirus immune response, T-cell analysis in this context may clarify important questions like if reinfections are possible, if other corona infections might provide immunity, or which role secondary diseases might play. Furthermore, analyzing T-cells in viral infections will allow a more exact prognosis on disease progression, opening thereby a more reliable assessment of action options, and may lead to better planning and therefore hopefully a faster return to a habitual life, resulting in also minimization of economic damages. But most importantly, we cover the whole antigen and virus-specific T-cell workflow to assist you in your research. Please also acknowledge the displayed disclaimer. With this, I come to an end of this presentation. Thank you very much for your attention and stay healthy.
Thank you, Dr. Stein, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you have any quantitative data on enrichment of other rare cells from blood, such as CTC or CFC?
Hello, everybody. So, no, at the moment we have not quantitative data in regarding to those cells. We actually here are focusing on the antigen-specific T cells that exhibit CD154 and CD137 enrichment. We usually at Miltenyi Biotec focus on on different CD markers to do so. So if you have a marker that you want to enrich, or if you want to enrich those cells by specific markers, then of course you can have a look if you have the respective enrichment kit, and there you will find for sure quantitative data.
Thank you, Dr. Stein. Next question, is it possible to stimulate enriched CD4 T cells with virus peptides without using APC cell?
So actually I'd say no. You need an antigen presenting cells, or at least some way of antigen presentation that presents the respective peptides to the T cells and provides an additional stimulus. So we always recommend to use PBMCs in this context, or to actually use isolated T cells together with, for example, monocyte-derived dendritic cells as APCs in a co-culture. This is a relatively clean system where you will get the reaction that you're looking for. Of course, it has to be autologous then.
Okay. What is the proportion of IFNG in enrichment CD4 T cell?
I guess you're saying how many cells in the enrichment are interferon gamma positive. So actually, I would need to look that up, what our average is. I mean, you can see it from the plots what it is, but actually, as I see, there are no numbers provided. For this, I would either recommend that you have a look on our website to find the respective kit, and then there for sure are plots displayed where we have a percentage, or you can simply call our MaxTech hotline and ask them for the specific block what the percentage was, and they will instantly answer you and give you the percentage.
Okay, thank you. Okay, let's see. Here's an interesting question. due to the relatively low affinity of autoantibody in autoimmune disease, do you have recommended Miltenyi kits that could be used to isolate or recognize antigen-specific T cells in autoimmune disease?
That's indeed an interesting question. So most of our data, and this is also the topic of this talk, was generated with virus-specific T cells. And there, the question is a little bit different, of course, because depending on which virus we are talking about, so CMV, for example, there still the cells are rare, but not as rare and with such a low affinity as the autoantigen-specific T cells. That's absolutely true. So I'd say this is something you have to try out. This would be absolutely my first recommendation. And in any case, for those types of cells, you need to use a very efficient pre-enrichment. So if you prepare your PBMCs, and then you stimulate with your autoantigen, and then you have a very, very rare population of reactive cells, then go via the pre-enrichment. It depends a little bit if it's CD4 or CD8 cells. If it's CD4 cells, then with CD154 pre-enrichment, you should be very well able to pre-enrich enough so that you, even though the cells are extremely rare, will be able to detect them. For the CD8 cells, either try rapid arte, modifying it by using CD137, or directly go on a cell sorter. With a cell sorter and appropriate amount of starting cells or PBMCs, then even with the rarest cells, you should be good.
Thank you. What is the difference between max quant analyzer and Tito?
The analyzer is a flow cytometer, and the Tito is a flow sorter. The max quant analyzer comes in different versions. So we have the max quant analyzer 10 with 10 optical parameters. We have the max quant analyzer 16 with 16 optical parameters. We have the max quant white that has a different laser setup for, especially good for cherry dye analysis. And we have the max quant X, which is a high throughput flow analyzer. So the analyzers, the max quants analyzers, the flow cytometers come in 4 versions. The max quant Tito for now is just 1 version, and only the Tito is a cell sorter.
Okay. Next question. Okay, let's see. Can we use LS column for negative isolation of CD4 T cells from PBMC?
Yes. So there are several approaches. It depends a little bit on the starting amount of cells that you have. If you're working with higher amounts, one kind of column is maybe suited. If you work with lower amount, another kind of column is suited. But generally, yes, you can do that. But I would always recommend that you go for the data sheet of the respective kits, because there are exceptions generally. So sometimes if you want to deplete, you would not use an LS column, you would use an LD column, for example. But the data sheet always gives you the recommendation of which column you have to use.
Okay, thank you, Dr. Stein. We have time for one more question. And the final question for today is, in analogy to cytokine, I apologize if I'm saying that incorrectly, capture assays, can you also capture antigen-specific antibodies on the surface of viable B cells?
No, unfortunately not yet. So this is a kit that we do not have in our portfolio yet. But it's a very interesting idea.
Thank you again, Dr. Stein, for for your wonderful presentation. Do you have any final comments for our audience today?
Just thank you all for listening. Stay healthy. And to you, Tracy, thank you very much for your support.
Before we go, I'd like to thank the audience for joining us today and again for their interesting questions. Questions that we did not have time for today and those that are submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. We would like to thank you, Dr. Stein, for your time today and your important research. And we would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. This webcast can be viewed on demand. Yeah, LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and friends who may have missed today's live event. Until next time, thank you again, everyone, and goodbye.